Good morning. Let us pray. Our lesson for today, it's all about Lesson 3, Reproductive Structure of Animal and Plants. Lesson outcomes. Number 1, describe the reproductive structure among animals. Number 2, describe the reproductive structures among plants. Animal reproductive structure. Human beings are classified as mammals along with goats, cows, dogs, cats, and mice. Mammals have external and internal sex organs. So, human beings are also classified the mammals, and also we have the external and internal. External genitalia that scientists use to identify whether the newly born individual is a male or a female. So, external genitalia also identify the newly born. Internal fertilization of the egg by the sperm occurs when there is mating of the male and female individuals. Gestation occurs inside the female uterus until a young is born. Among birds such as chicken and rooster can be identified by the comb on its head and its prominent tail feathers. So the chicken and the rooster, dito mo sila ma-identify if male or female. Here is the example of chicken and rooster, yung picture na pinakita ko. Cloaca is an opening in the bird through which sperm and egg pass through. Snakes and lizards have no external organs to help scientists identify male from female, though they exhibit variation in appearance. So the snakes and lizards have no external organs. Frogs have paired gonads and oviducts that are found inside the body, which are connected to the cloacae or plural of cloaca. Paired gonads means the primary reproductive organs are the testes in the male and the ovaries in the female. Oviduct means are paired tubes extending from each ovary to the top of the uterus. Reproduction in fish and amphibians. The female fish has a paired ovaries that produce eggs that are released through oviducts to the uterus or the body cavity. The male fish has paired testes that produce the sperm. So the female fish has paired ovaries. Paired ovaries means one of the pair of female glands in which the egg form and the female hormones estrogen and progesterone are made. Paired testes means one of the two egg-shaped glands inside the scrotum that produce sperm and male hormones. Here is the example of earthworm reproductive system. Next is reproduction in insects. Atypical modes of reproduction. Parthenogenesis, development from unfertilization eggs. Petagenesis, loss of adult stage and development of young within immature. Neoteny, immature looking adult. Mating. Monogamous, when females of some species are essentially mate once. Polygamous, when female insect mates several to many times. Hydra. In the case of Hydra, a coelenterate growing in fresh water, a new animal is produced by a bud from its main body, which eventually grow and separate from the parent body. Some organisms that reproduce sexually may also reproduce asexually. Among staghorn corals, broken segments of the colony may grow into a new colonies. A sea star that loses some of its arms may grow or regenerate the loose parts to become a new individual. Plant reproductive structures. Most plants that you see around have flowers, which are the sex organs of these plants. Among non-flowering plants like in the pine trees, an individual produces male and female cones. In ferns, the spores formed in the leaves germinate into heart-shaped plants that contains in the sex cells. Mosses have spores that germinate to young moss plants, but grow into male and female that produce the sex cells. There are the plant's parts like leaves, stems, and roots that give rise to new plant. Structure. As a plant's reproductive part, a flower contains a stamen, a male flower part or pistil, female flower part or both plus accessory parts such as sepals, petals, and nectar glands. The stamen is the male reproductive organ and consists of a pollen sac, anther, and a long supporting filament. Flower structure. A typically flower has a four main parts or whorls: the calyx, corolla, androecium, and gynoecium. The outermost whorl of the flower has green leafy structure known as sepals, which are collectively called the calyx and help to protect the unopened bud. The second whorl is comprised of petals, usually brightly colored, collectively called the corolla. The number of sepals and petals varies depending on whether the plant is monocot or dicot. Together, the calyx and corolla are known as the parent. The third whorl contains the male reproductive structure and is known as the androecium. The androecium has stamens with anthers that contains the microsporangia. The innermost group of structures in the flower is the gynoecium, or the female reproductive component. The carpel is the individual unit of the gynoecium and has a stigma, style, and ovary. A flower may have one or multiple carpels. Structures of the flower. The four main parts of the flower are the calyx, corolla, androecium, and gynoecium. The androecium is the sum of all the male reproductive organs, and the gynoecium is the sum of the female reproductive organs. If all four whorls are present, the flower is described as complete. If any of the four parts are missing, the flower is known as incomplete. Flowers that contains both an androecium and a gynoecium are called perfect androgynous or hermaphrodites. There are two types of incomplete flowers. Staminate flowers contain only a androecium, and carpellate flowers have only a gynoecium. Staminate and carpellate flowers. The corn plant has both staminate male and carpellate female flowers. Staminate flowers, which are clustered in the tassel of the tip of the stem, produce pollen grains. Carpellate flowers are clustered in the immature ears. Each strand of silk is a stigma. The corn kernels are seeds that develop in the ear after fertilization. Also shown is the lower stem and root. Superior and inferior flowers. The lily is a superior flower which has the ovary above the other flower parts. Foxy is an inferior flower which has the ovary beneath other flower parts. Male gametophyte. The male gametophyte develops and reaches maturity in an immature anther. In the plant's male reproductive organs, development of pollen takes place in a structure known as the microsporangium. The microsporangia usually Below are pollen sacs in which the microspores develop into pollen grains. Microsporangium shown is a cross-section of an under a two developmental stages. The immature under top contains four microsporangia or pollen sacs. Each microsporangium contains hundreds of microspore mother cells that will each give rise to four pollen grains. The tapetum supports the development and maturation of the pollen grains. Upon maturation of the pollen bottom, the pollen sac will split open in the pollen grains. Male gametophytes are released. In these scanning electron micrographs, pollen sacs are ready to burst, releasing their grains. Each pollen grain has two coverings, the axen thicker outer layer and the intine. The axen contains sporopollen, a complex waterproofing substance supplied by the tapetal cells. Sporopollen allow the pollen to survive under unfavorable conditions and to be carried by wind, water, or biological agents without undergoing damage. Pollen grain structure. Pollen develops from the microspore mother cells. The mature pollen grain is composed of two cells, the pollen tube cell and the generative cell, which is inside the tube cell. The pollen grain has two coverings, an inner layer, intine, and an outer layer, exine. The insect scanning electron micrograph shows Arabidopsis lyrata. Female gametophyte. Embryo sac. The overall development of the female gametophyte has two distinct phases. First, in the process of megasporogenesis, a single cell in the diploid megasporangium undergoes meiosis to produce four megaspores, only one of which survives during the second phase. Megagametogenesis, the surviving haploid megaspore undergoes mitosis to produce an ethnoculate seven-cell female gametophyte, also known as the megametophyte or embryo sac. The polar nuclei move to the equator and fuse, forming a single diploid central cell. This central cell later fuses with a sperm to form the triploid. In those sperm, three nuclei position themselves on the end of the embryo sac, opposite the micropyle, and develop in the antipodal cells, which later degenerate. The nucleus closest to the micropyle becomes the female gamete or egg cell, and the two adjacent nuclei develop into synergy cell. The synergies help guide the pollen tube for successful fertilization, after which they disintegrate. Once fertilization is complete, the resulting diploid zygote develops into the embryo. The fertilized ovule forms the other tissues. Double-layered integument protects the megasporidium and later the embryo sac. The integument will develop into the seed coat after fertilization, protecting the entire seed. The ovule wall will become part of the fruit. The integuments, while protecting the megasporidium, do not enclose it completely but leave an opening called the micropyle. The micropyle allows the double-layered integument protects the megasporidium and later the embryo sac. The integument will develop into the seed coat after fertilization, protecting the entire seed. The ovule wall will become part of the fruit. The integuments, while protecting the megasporidium, do not enclose it completely, but leave an opening called the micropyle. The micropyle allows the pollen tube to enter the female. Thank you for listening.